All right, let's see. Can everybody hear me? Great. Hi. Seeing some familiar faces and some new faces. Thank you so much, Ed, for the nice introduction and welcome. Yeah, nice to meet you all for those who I haven't met yet. This is my first time giving a talk for Center of Research, and I'm really looking forward to getting to know many of you in the coming years as I'm growing my lab here at UBC. And so today, I thought I would give sort of an overview of the work that I have done so far that has really led to some of the projects that are ongoing in the lab right now, in my independent lab right now, and sort of the future directions of where we're going and some exciting new areas that we're sort of venturing into in the lab. The title of my talk is Interplay between endogenous retroviruses in inflammation. And I'd first like to, start this talk by acknowledging that we are all here working, and I personally also live on this land of the Musqueam people. And I usually like to reflect on the Musqueam culture vision and mission and their values as I think about the land that I am living on and the land that I'm working on. And I really especially value their values, the values of respect, pride, inclusiveness, honor, and shared responsibility. And I try to carry this on through my research and my life here as well. All right, so with that, I like to get started, and I like to introduce you to the current members of the Tokuyama Lab, some of whom are sitting in the audience here today, and so you might be able to meet them afterwards. We're a growing lab. We have a number of new students in the lab, PhD students, master's students, as well as undergraduate students from various departments at UBC. And we are very much a endogenous retrovirus-centric lab. We get to talk about ERVs all the time, which is the most exciting part of my day. And I'm going to share a little bit about sort of what everybody is working on sort of through the lens of our research interest. All right. So really the bottom, sort of fundamentally what we're really interested in through our work is trying to understand heterogeneity in health and disease. And so as we all experienced and really noticed during the COVID-19 pandemic, really disease development is a very heterogeneous process. A lot of people can become infected with a pathogen and completely be fine, have asymptomatic, no symptoms, and fare well, and while others really succumb to infection and have severe disease. And really, this range in disease outcomes and disease manifestation can be attributed to many factors that many of us are familiar with and many of us work on here. That could be genetic, microbial, environmental, hormonal, even demographic differences can really contribute to this larger overall heterogeneity in health and disease. And in my lab, we're really interested in the contribution of viral elements that can contribute to this heterogeneity in disease. And so as a whole, the bigger umbrella that we're really studying under is this concept of the human virome. Many of you are probably familiar with the microbiome, but perhaps you may not have heard of sort of the virome or be less familiar with this. But really, we are composed of many, many viral elements in our bodies. This was sort of this is a recent publication that has really shown that throughout our organ system in various tissues throughout our body, we actually have many, many bits of pieces of viruses that reside within us. And we really carry these viral elements with us long-term for the length of our life. And how does that impact our health, right? These viral elements can, in fact, come from viruses that are acquired during life. So exogenous viruses that you may be exposed to in your childhood. It could be viruses that are exposed to more recently. And there are also viruses that are genome encoded, viral elements that are genome encoded. And all of these are really, again, lifelong infections. So we're constantly, interacting with these viral elements and seeing these in this persistent and chronic interaction between the immune system and these viral elements can really potentially contribute to the heterogeneity that we see in disease outcomes in health and disease, right? And so in my lab, we're really particularly interested in this the genome encoded viral elements that are part of the viral metagenome, and these are called ERVs, or endogenous retroviruses. And so what are these endogenous retroviruses? And maybe some of you have come to some of my other talks and maybe are becoming more familiar with it and hopefully more excited about them. But for those of you who are not familiar, endogenous retroviruses are really relics of ancient retroviral integration events. They're thought to have originated from an exogenous retrovirus infection, much like HIV. And these exogenous retroviruses underwent infection of a target cell, reverse transcribed its RNA genome, and then led to integration. And those proviruses that actually integrated into the germline and became fixed and sort of inherited over time are what we are now calling endogenized or endogenous retroviral elements. These have been carried over through evolution and have now are taken residence in all of our DNA. And it is thought that there have been sort of peaks in spikes in integration events through evolutionary time. I realize that these seem like very long time ago, but there are ways in which we can date some of these endogenous retroviral elements and predict their integration time. And these are sort of the predicted timelines for which we've had major peaks of integration events. So 80 million years ago, roughly 50 million years ago, 7 to 5 million years ago. And we also have more recent integration events under 1 million years ago that are really more Homo sapiens specific integration events. And it turns out that when the human genome was initially sequenced in 2001, this was a large work that was done by a large consortium of the Human Genome Sequencing Consortium that was led by Eric Lander. It was uncovered that 8% of human genome is actually composed of these LTR elements or viral elements, LTR retro element sequences. And that is in fact 4 times more than the 2% of the genome that encodes for proteins. And these collectively, these endogenous retroviruses belong to a family of repetitive or transposable elements that also consist of DNA transposons, lines, and sign elements. And what's something that I keep in mind as I carry out my research is a quote in this paper that really says, repeats were often described as junk prior to sort of the genome sequencing events, but however, they sort of represent this extraordinary trove of information about biological processes. And I really kind of take that to heart. It's a bit like a treasure hunt to go in there and identify novel viral sequences and figure out what they do. And really, it's also not only sort of a treasure trove of information, but it's also important to understand and get insight into evolutionary events and forces that have shaped our genome and transcriptional regulatory mechanisms that regulate all of the biological and physiological processes that we're familiar with today. All right, and these transposable elements really that ERVs belong to are also conserved throughout most eukaryotic genomes, right? This is not sort of just a human specific phenomenon, that these elements are in fact found in all species, most eukaryotic genomes. And some of you may be familiar with the fact that Dr. Barbara McClintock won the Nobel Prize in 1983 for her work on transposable elements in understanding how the phenotypes that she found in maize actually can be attributed to transposable elements. And so ERVs belong to this larger group of transposable elements, and many in the field are really interested in how these transposable elements, including ERVs, are really regulating physiological processes. And as a viral immunologist, my lab is really interested in, so what do these sequences do to our immune system, right? How does it affect our immunity? How does it affect inflammation? How does it affect disease? And so we're really focused on sort of the viral components of these elements that can contribute to immune modulation and disease development. And so we have to kind of think about what these viral elements actually are in order for us to understand how they might impact our immune system and how they might modulate health and disease. And so just to kind of come back to the basics, these retroviral sequences, going back to herbs and these retroviral sequences, initially when these retroviral sequences integrated into our germline, they integrated as proviral sequences or proviral herbs, and these are full-length proviral sequences that are flanked by a 5' and 3' LTR and open reading frames that are responsible for packaging, for integration and packaging and replication of progeny virions. However, over evolutionary time, there have been a lot of deletions, homologous recombination between the two LTR sequences, the 5' and 3' LTR sequences, that have now resulted in sort of a scattering of varying types of sequences that belong to the endogenous retroviral family in the genome. So some of these sequences take the form of solo LTRs, that as a result of recombination between the 5' and the 3' LTR sequences. And then some are sort of internal sequences that have just been sort of left behind through evolutionary time. However, there are still, in fact, some proviral sequences that still remain in the genome, and we're very much interested in this. And so what do these sequences actually do? And what do we know about them? We know that some of these solo LTRs are really important factors in regulating gene expression, genes that are nearby, and they serve as really potent cis regulators. We also know that, on the other hand, proviral sequences can actually lead to transcription of polyadenylated mRNA and generate viral proteins or even extracellular vesicles that in and of itself can have functionality. And in these herbs and herb products, such as viral proteins and some of these extracellular vesicles have actually been shown to be very important in basic physiological functions like embryogenesis and even placental development. However, more recently, there's been a whole list of sort of diseases that these herbs have been implicated in. And there's a large list that I've just kind of listed here. This is by no means an exhaustive list, but you can see that these herbs have been really implicated in many diseases, including neurodegenerative diseases, autoimmunity, and even cancer. And so these herb elements that are really that all of us kind of encode in our genome have been really co-opted both for beneficial purposes, for really basic physiological purposes, but they can also cause detrimental. They can be implicated in detrimental effects like disease. And so, how does this all work, and what are sort of the mechanisms, and in sort of understanding how they might modulate the immune system? A handful of sort of ideas have been, and studies have shown that these endogenous retroviruses can potentiate immune activation, and that can be one way in which they can contribute to disease. And so these endogenous retroviruses that encode these viral elements can be transcribed, and upon transcription, they can generate RNA intermediates that can actually stimulate cytosolic innate sensors like RIG-I, MDA5, and cGAS, as well as endosomal sensors like TLR7. And these have been shown to lead to activation of type 1 interferons. These proteins have also, by themselves or in the context of extracellular vesicles, have also been shown to engage with TLRs or toll-like receptors that are expressed on the cell surface and engage the signaling pathway that can lead to pro-inflammatory cytokine production. We've also seen that there's quite a bit of immune surveillance of these ERV proteins. We've observed that in different disease contexts, antibodies can be derived against these endogenous retroviral proteins and can mediate inflammatory processes. And there's also been T cells that have been pulled out in cancer patients that are really specific to these endogenous retrovirus peptides, and some of them have also been shown to be serving as super antigens. And so there's sort of this plethora of potential for these endogenous retroviruses to modulate the immune system. And yet, really, there are still many, many questions to be answered in this area. And so, for example, some of the questions that sort of dictate our research are, what are the consequences of this chronic interaction, right? Not just sort of short-term interaction, but this lifelong interaction between herbs and the immune system. And how do these interactions actually contribute to immune homeostasis? And then in turn, how does that contribute to inflammation and even disease development or progression? Another is how are herbs actually being regulated? How is herb expression regulated? And what is the immune surveillance? What are the immune surveillance mechanisms against herbs beyond sort of some that we have already seen in various studies? Are there other mechanisms that we have developed or devised, our immune system has devised to be able to survey, to have surveillance against these viral proteins or viral products? And how do these herbs interact with other microbial communities? Of course, this is part of a viral, the herbs are part of the virome, but the virome doesn't act by itself. They also interact with the microbiome and other microbial communities at various mucosal sites. And so how do these sort of interkingdom interactions actually then contribute to immune modulation? And then finally, ultimately, we're really interested in using all the basic knowledge that we come up with to understand and see if we can use herbs and leverage them for therapeutic purposes. All right. And so sort of based on all of these questions that we're really interested in, my lab is really divided into sort of three major areas of research. One is really continuing to understand what these proviral sequences are. What are these proviral sequences? We're still starting to beginning to understand sort of the genomic annotations and genomic sequences, but we're really far from complete in this understanding. We would like to understand how they're expressed, what they actually produce, all of these endogenous retroviral sequences within the genome, what are they actually making, and what's the transcriptional regulation of these herbs in the context of the immune system. And in turn, how do these herbs that are expressed and the products that are expressed from these endogenous retroviruses modulate our immune system? I gave you sort of examples of how our immune system can be activated, potentiated by these endogenous retroviruses, but again, this is sort of just the tip of the iceberg. Are there other functionality that herbs can serve in immune cell functionality and immune homeostasis? And in turn, how do these interactions then contribute to whether it be beneficial effects of our immune system like pathogen clearance or detrimental outcomes of these interactions, such as development of chronic inflammatory diseases like autoimmune diseases? And so I'll give you a little bit of a snippet of highlights of what we do in each of these areas. And hopefully you can sort of by the end of this talk get an appreciation for sort of the breadth of our research program here. And so to begin with, I like to introduce our bread and butter. This is something that I had developed during my postdoc when I was a postdoc. And really, this is a computational bioinformatic tool to be able to actually map proviral herb sequences in RNA sequencing data. And so this was at the time when I first started this project, was still very much new. RNA sequencing had just been sort of emerging and starting to be really become common. This is still bulk RNA-seq, not even single-cell RNA-seq. And we were able to do this RNA sequencing, but we really didn't have a handle on how to actually analyze and mine those RNA sequencing data sets for endogenous retrovirus expression. And part of it is the fact that these are repetitive sequences that are really hard to sort of dissect and investigate. And so one of the things that I initially started with was developing this herb map. And really, herb map consists of a database of proviral herbs that have been annotated by many others in the field. And this had not been done before. Even the genome annotation that you may or may not be currently using through UCSC is not fully annotated in terms of these proviral herbs. And so this was an important component that we actually had a database for these proviral herbs. And then we also had to sort of think about how we're going to deal with these repetitive sequences and how to analyze these RNA sequencing RNA sequencing data for ERV transcription. And so we developed this pipeline, and what we were able to do with this is to really be able to see that these ERVs are in fact dynamically expressed in various conditions. This is just a few examples in which we've been able to use ERVmap to look at sort of dynamic expression of endogenous retroviruses. For example, if we take sort of your standard cell lines that many of you may be familiar with working in the lab, you can see that distinct cell lines express distinct sets of endogenous retroviruses, and the cell types are color-coded in different colors. And so you can see that different cell types actually have different and unique ERV signatures. And this is sort of the first time we had actually been able to see that. We can also use this in other transcriptome data sets and see differential expression of these ERVs, right? And so we had done this for a cancer data set as well as an autoimmune data set. And so we were able to kind of show that this is a bioinformatic pipeline that we can actually utilize to then further investigate more mechanistically how these ERVs are actually involved in disease and in inflammation. And so I'll give you an example of a story that was published still from when I was a postdoc, where we had utilized this pipeline to really be able to understand and uncover novel sort of inflammatory pathways that were involved in autoimmunity in the context of lupus. And so just to give you a quick overview on what systemic lupus erythematosus is, which is the disease that I'll be focusing on for the next 10 minutes or so. This is a systemic autoimmune disease. It affects about 1.5 million Americans, roughly 5 million people worldwide. And really this is a systemic autoimmune disease that affects a number of people, yet there's only 2 FDA-approved biologics available. One was approved in 2011, that's a belimumab anti-BAFF antibody, and anifrolumab, which is an anti-interferon alpha antibody, was recently approved in 2012. But really, there's only a handful of biologics that are available for this disease. And one of the biggest challenge with this disease is that it's very heterogeneous. So there is different disease manifestation within the lupus population, and it makes treatments very, very challenging. And not only and part of the reason why this is a heterogeneous disease manifestation is because it affects many different organs throughout the body. And lupus patients come in with a variety of clinical symptoms that affect different tissues. And so that also kind of adds to the complexity of this disease. There are many contributing factors that have been attributed to this disease, including genetic factors, epigenetic factors. This is a female dominant disease. There's a 9-to-1 female dominance in sort of disease incidence. And there's also racial distributions that where Asians and African Americans are actually there's a higher incidence of lupus in Asians and African Americans than Caucasians, and a variety of number of other factors contribute to this. And so why am I talking about this when we're talking about ERVs, right? Well, we had known from decades of work that ERVs had actually been implicated in lupus pathogenesis. And there had been a handful of studies that had shown that in these patient cohort, these lupus patients, there were immune complexes that have been identified that were targeting ERV proteins, specifically ERV-P30. And there had also been sort of antibodies against the GAG protein of endogenous retroviruses that had been detected in lupus, and also T cells that had been that express this ERV-HRES that had been heightened and elevated in lupus patients. And so they had been implicated in lupus for a while, for decades. In addition to this, all models of sort of mouse models of lupus pathogenesis, MRLs, BXSB, and ZBWs, all of these actually have highly elevated ERV envelope GP70 proteins in the serum, and they're accompanied by antibodies that are also against these GP70 proteins that are elevated in these mice. And people had seen immune complexes of these GP70 immune complexes deposited in the kidney. And so again, there had been implication that ERVs had been potentially contributing to lupus pathogenesis, but really there hadn't really been a full analysis of how, mechanistically, how ERVs can potentially contribute to the systemic systemic inflammation, and more broadly, how does that contribute to sort of the clinical disease and the clinical manifestation that we see in lupus patients. And even though there had been some indications of antibodies against ERV proteins, really didn't have a good target of where these antigens were coming from and which ERV they were coming from. And so, based on sort of our interest and using sort of the newer technology of RNA sequencing and the ERV-map pipeline that we had, we decided to apply our ERV-map pipeline to understand lupus patients and ERVs and ERV dysregulation in lupus patients more broadly, and then ask, what are the mechanisms that may contribute to inflammatory processes in this disease. And so this is just one of the, and I'll just give you some highlights from this paper, but we had seen that in the whole blood of these lupus patients, and we had actually have another cohort that we also analyzed where we also further saw similar results where lupus patients have elevated ERV expression. And so this is looking at, SLE in patients in red and healthy individuals in blue. You can see in red all of these endogenous retroviral loci, which are indicated in each of these rows are distinct ERV loci. These are all elevated in these lupus patients. And this is sort of beyond sort of the two ERV loci that had previously been implicated in SLE. And so, we were able to kind of start with this larger pool of endogenous retroviruses to really investigate how these are important or involved in disease development. We further saw that the expression of these endogenous retroviruses correlated with clinical measures like autoantibodies. So anti-nuclear antibodies, anti-double-stranded DNA antibodies, these are classical hallmarks of disease in lupus patients. And these both correlated with the expression of endogenous retroviruses in these patients. And so given that these ERVs are elevated, we started to sort of narrow down into what type of ERVs may be important in terms of inflammation in this patient group, and we narrowed down out of sort of all the 100 or so ERVs that we had seen elevated. We narrowed down on the specific family of ERVs called the ERV-K family of ERVs, and the ERV-K family, or the human ERV-K family of ERVs, these are the youngest endogenous retroviruses that we know in our genome. So as I mentioned earlier, there are sort of recent integration events that happen specifically in Homo sapiens, and ERV-K is really one of the younger ones. So these are were integrated, we think, less than 1 million years ago, and because these are one of the younger integration or the youngest ERVs, many of them are intact sequences, intact proviral sequences, and they still encode viral proteins. They've been shown to form extracellular vesicles in various contexts, and it's been implicated in many diseases, including ALS, cancer, as well as infection. And recently, it's really been thought of as sort of promising targets for various therapies in different diseases. And so kind of focusing in, zooming in on this family of ERVs and pulling these out of our database. So in our database, we have over 3,000 proviral ERV sequences. Out of those, roughly 87 of them belong to the ERV-K family, and 12 of them actually encode an intact envelope sequence. And this was interesting to us because we had known from the MRL mouse models that the GP70, the ERV envelope proteins, are highly elevated in the mouse models of lupus. And so we really wanted to see if something similar was going on in lupus patients, and really narrowed in on these envelope protein encoding ERV-Ks. And so when we looked at the expression of these 12 envelope encoding ERV-K family members and compared them between SLE and healthy, we found 4 of these ERV-K loci that were highly expressed in SLE, and they were significantly elevated compared to healthy controls. And the top one was this ERV-K102 locus, and that's encoded in chromosome 2. And this was really the highest expressed and significantly elevated in lupus patients, and we sort of started to narrow in on, potentially what this particular locus encodes and how that could contribute to inflammation. And so this is the ERV-K102. From going for the next couple of slides, I'll be focusing specifically on this. And so one of the things that we want to understand was, so what about these ERV-K102s? Do they actually have any correlation with any inflammatory signatures in these patients? And so and similar to other autoimmune diseases, lupus patients have really highly elevated interferon signature. This is a classic feature of these patients. And again, and as I mentioned earlier, this is a female dominant disease. And so what we had noticed was that in this cohort, ERV-K102 was highly elevated in females compared to males. And this was a significant difference where the females had significantly elevated ERV-K102, but males did not. And the ERV-K102 expression significantly correlated further with the interferon signature metric, or the ISM. And so those individuals that had a high ISM had higher expression of ERV-K102 compared to those lupus patients that had lower amounts of interferon signature, right? So there was this direct correlation that we saw with ERV-K102 expression and interferon, especially in female patients. And so we isolated the envelope protein, or generated recombinant protein of these envelope protein of this envelope from ERV-K102, and asked whether these patients have developed autoantibodies against them, because this is an autoimmune disease, and the autoantibodies are really classic features of these patients. And so we generated a recombinant protein against K102 envelope, or K102 envelope recombinant protein, and used both ELISA and Luminex assay to be able to assess whether there is any difference in the amount of autoantibodies that might be reactive against K102. And we actually didn't see a big difference between healthy and SLE patients. So both healthy individuals and SLE patients had similar levels of antibodies against these ERV-K102 proteins. However, what we noticed was that when we actually look at within the SLE patient groups, the titer, there's clearly a range in ERV-K102 antibody titers within this patient group. And if you actually look to see within this patient group, do the differences in antibody titers actually correlate with differences in inflammatory signature? We saw this positive correlation between sort of the amount of anti-ERV-K antibody that you have, which is observed in this on the x-axis here, and the amount of interferon signature genes that these individuals express. So there was a direct correlation between sort of higher amounts of antibody and higher amounts of interferon signature, interferon signature gene expression in these patients, suggesting that even though sort of there's no large difference between the total amount of ERV-K102 antibodies that are present between healthy and SLE individuals, there seem to be some kind of functional differences between these groups. And so at this point, we collaborated with Galit Alter from the Reagan Institute and her postdoc, Bonnie Gunn, and really decided to assess whether these antibodies are functionally distinct. And Galit has this really amazing high throughput antibody effector functionality assay. And so we really took advantage of this with this collaboration. And what we had actually pulled out was that although, again, the amount of antibody against these ERV proteins were not distinct between healthy individuals, they seem to be functionally different. And so one of the differences that we found was based on this assay that we did, looking at the ability of neutrophils to be able to phagocytose immune complexes. So this is an antibody-dependent neutrophil phagocytosis assay, which is one of these antibody effector functionality assays that are part of sort of this high throughput system. And so how this works is that you generate immune complexes with your antigen of interest, and this, in our case, which is it's an ERV-K102 envelope. We conjugate them or make an immune complex with either healthy serum or SLE serum, and then we feed these immune complexes to healthy neutrophils and quantify the amount of immune complexes that have been phagocytosed in neutrophils. And what we had found was that interestingly enough, that when you make these immune complexes with SLE patient serum or IgG that's purified from SLE serum, these antibodies against these ERK102 envelope proteins actually seem to have a higher ability for enhance the ability of neutrophils to phagocytose these immune complexes. And this is not sort of a general phenomenon of SLE antibodies. In fact, if you just look at sort of a common antigen like influenza HA or even tetanus toxin antigen, these antibodies don't seem to have any differences in terms of its functionality. And so we're really seeing sort of this ERV-specific enhancement of these of neutrophil phagocytosis by these antibodies. And we further characterize sort of how this might be happening. And we performed an Fc gamma receptor binding assay for these immune complexes to really assess, who's sort of responsible for uptaking these immune complexes. And so this again is part of this antibody effector functionality sort of assay that Galit has set up, where we look at Fc gamma receptor binding, recombinant Fc gamma receptor binding to the immune complexes that we provide. So in our case, we have ERV-K102 immune complexes from SLE patients, and then we see how well they bind to various Fc gamma receptors that were known to be expressed on neutrophils. And what we found was that the amount of binding that you see to Fc gamma receptor 3b in particular had a positive correlation with an enhanced neutrophil phagocytosis that we had observed. So the ADMP, the antibody-dependent neutrophil phagocytosis, was positively correlated with the amount of these immune complexes that bound to Fc gamma receptor 3b. And we further blocked this Fc gamma receptor 3b, which is a low affinity Fc gamma receptor that's highly expressed on neutrophils in this phagocytosis assay. And we really observed that the presence of anti-CD16, which is an antibody against Fc gamma receptor 3, reduced or ameliorated this enhancement in antibody-dependent neutrophil phagocytosis of these immune complexes, really suggesting that these anti-ERV-K102 immune complexes formed by SLE antibodies are being phagocytosed by neutrophils via the Fc gamma receptor 3b that's expressed on their surface. And we further asked whether that actually has functional consequences on neutrophil activation. And so neutrophils are known to sort of become activated and protrude their intracellular DNA and form these things called neutrophil extracellular trap or nets. And what we had found was that when you feed these neutrophils, these immune complexes, these ERV immune complexes from SLE that are generated with SLE antibodies, they actually lead to heightened net formation compared to immune complexes that we feed to immune complexes that are generated with healthy IgG that we feed to neutrophils, where we don't see this net formation. And so we were able to sort of, and this is sort of quantified here on the right. And this is important for lupus pathogenesis because nets are really a key component, a driver in interferon responses due to the fact that these extracellular DNA that it now protrudes can now be ligands for further anti-double-stranded DNA immune complex formation, leading to activation of cells like dendritic cells, and then leading to more interferon and cytokine production. And so we think that potentially the ERV-K immune complexes that can be formed in this disease can kind of potentiate this whole pathway by activating upstream these can contributing to this upstream neutrophil activation and then sort of propagation of this interferon response. And so this is sort of a highlight of this work. We were really able to see differential ERV expression, using our ERV map, comparing healthy and SLE patients. We are able to see that despite the fact that both healthy and SLE individuals have similar amounts of anti-ERV-K IgG, that the ERV-K antibody within SLE patients seem to contribute more towards the ISG signature, the interferon signature. And potentially we think this is through this enhanced antibody-dependent neutrophil phagocytosis that can lead to net formation and further inflammatory consequences. And so we were lucky enough that this was received very well by the community. We got highlighted in the JEM Clinical Immunology Collection in 2021, and our lab received the lupus, not my lab, but my former lab, received the Lupus Insight Prize from the Lupus Research Alliance for this work. And sort of kind of building off of this work, we now have a project in the lab with collaboration with Laura Avgan from BC Cancer, where we're trying to design chimeric autoantibody receptor T cells against ERV envelope proteins to see if we can deplete these, the sources of these ERV antibodies, and potentially that can lead to reduction in inflammatory consequences. So stay tuned in this area. So I don't know what time it is. Okay, so sort of switching gears a little bit. This is sort of a nice sort of way for us to be able to kind of go from, bioinformatic analysis to be able to do some functional characterization and even get some clues into how these endogenous retroviruses can contribute to inflammation in disease in a clinical setting. And so we, like to use this sort of process to be able to understand other disease types and other inflammatory diseases. And so just kind of switching gears a little bit. Again, we're using sort of this pipeline to be able to ultimately understand how these ERVs are contributing to whether it's detrimental or beneficial effects. And so to this end, we have, I like to share a little bit of one of the newer projects that we have in the lab, which is part of a sort of a bigger goal for us to be able to use this type of analysis that we're using to really map human endogenous retroviruses and disease. And we're hoping to be able to use a similar approach as what we did in the lupus patients to be able to sort of identify novel links between ERVs and inflammatory processes and disease by sort of doing a really comprehensive analysis. And so this is being spearheaded by Bessie Wang and Thomas Deckers in the lab. And I'll show you a little bit about one disease that we've been focusing on most recently, which is, of course, COVID-19, which is sort of what was how we started our lab. We started in the middle of a lockdown, and COVID-19 was clearly on our minds. And so we really wanted to use this sort of pipeline to kind of get our handle on how ERVs are involved in COVID-19 disease. And so what we know is that a lot of viruses, not just SARS-CoV-2, but in the past we had known that a lot of viruses are able to transactivate ERVs. In fact, HTLV, HCV, even HIV and HSV1 have been shown to transactivate ERVs. What that actually ends up doing or what the consequences, no one had ever shown. But I think this had been a phenomenon that people had seen. And in fact, when the pandemic started, there were some groups that looked at ERV expression in COVID-19 patients, specifically looking at ERV-W envelope and ERV-K, and found that ERV-W envelope protein is high, expressed, elevated in COVID-19 patients in various immune cell types. And even for ERV-K, there had been this differences in ERV-K expression in depending on the severity of the disease. And in fact, that contributed to sort of prediction of the, whether you had better survival, worse survival in disease. And so given this sort of precedence for ERVs in viral infection and even in COVID-19, we wanted to kind of use our approach to start to understand how we could contribute in this area. And so Bessie, Thomas, and Eric had been spearheading this work, where they've been looking at RNA sequencing data sets that have been published from various COVID-19 cohorts. And here we're really focusing on two different cohorts. The first cohort is looking our cohort of patients that are COVID-19 and then compared COVID-19 positive or SARS-CoV-2 infected and had COVID-19 symptoms and compared to healthy individuals. And so this cohort has about 10 healthy individuals, 44 severe COVID-19 patients, and these are whole blood samples compared to cohort 2, which is non-COVID versus COVID infection comparison. And these are sort of the stats. And again, these are whole blood RNA sequencing samples. And we kind of applied a similar method to try to identify ERVs that are dysregulated in COVID-19. And so what have we found so far? Certainly this is an ongoing work, but so far, Bassi, Thomas, and Eric have found a set of herbs that are elevated in COVID-19 patients. And these are sort of highlighted in these volcano plots. These are the herbs, distinct proviral herb loci that are highly upregulated in COVID-19 patients compared to healthy controls. And you can see even in this heat map that if you take some of these significantly elevated herbs and sort of plot them across all the different patient data sets that we have, that the COVID-19 patients really have elevated levels of, many of the COVID-19 patients have elevated levels of these herb loci. And we can even look at, what actually are these herbs that are being expressed. We can say this particular herb is up and this particular herb is down, but really we wanted to further dissect, what part of the herbs are actually expressed, and that can also inform us to potential mechanism for how these herbs might be contributing or involved in disease. And so these are just 3 representative herbs that are highly elevated in these COVID-19 patients, and we're taking sort of the 3 highest expressed herb patient samples from both COVID and healthy, and mapping the read coverage from these RNA sequencing data sets across 3 different herb loci that are indicated here. And what you can see is that really the entire span of the proviral locus is highly expressed in COVID-19 patients. And this is compared to green in healthy individuals in green. And so really what we think is, what we can really see now is that not only are these herbs elevated and we can pick up reads that come from these proviral herb loci, not only sort of do these reads come from that, but really the entire proviral locus is actually transcribed and expressed. All right, and so how does that relate to potentially other inflammatory consequences in these patients? So what we then did was correlate the expression of these herbs and upregulate cellular genes that are observed in these COVID-19 patients. And Bessie found these nice clusters of herbs and genes that really positively correlated with each other, and pulled out 4 that were interesting, and pulled out here depicted in this diagram on the right. So there's 4 clusters that we found where there are proviral loci that are elevated in COVID-19 patients that are associated with distinct sets of cellular genes, again, inflammatory genes that are elevated in COVID-19 patients. In particular, what stood out to us was this 4th cluster, this cluster 4. And the reason why that stood out to us was because many of the genes that are within this cluster that involves 2 herb loci, ERV111 and ERV943, all of these sort of genes are associated with neutrophil activation markers, or markers of neutrophil activation. And in fact, many of these COVID-19 patients who had severe COVID-19 patients display heightened neutrophil activation as part of the signature of disease. And so we were really interested to kind of see why that was the case, that these particular sets of herbs are significantly correlating with these neutrophil activation markers. And so we then turned to the other cohort that we had, the second cohort, which is where we're comparing sort of COVID and non-COVID. And we found that those same sets of neutrophil activation genes are also directly significantly correlated with ERV943 and ERV111 in those COVID-19 patients as well within that cohort. And again, these herbs are also highly induced in elevated in COVID-19 specifically, but not in patients who display COVID-like symptoms, but were not COVID. And so we really think that there's an interesting link between these two distinct herb loci and neutrophil activation that's specific to COVID-19. And so we're kind of pursuing a number of questions here: which cells are expressing these herbs, and how are herbs contributing to inflammation, and are they being upregulated as a consequence of inflammation, or are they actually contributing to inflammation? We're also interested in understanding if these herbs can serve as markers of disease severity or therapeutic targets, and these are many of the questions that we are trying to answer right now. One really cool thing that Bessie recently found, this is very preliminary, but I think this is really neat, and so I thought we would show this. We've been mining long read sequencing, so Oxford nanopore data sets, to really be able to see if this these ERVs are transcribed as a full-length transcript. And that gives us a little bit more confidence as to what we're dealing with here. And so some of these ERVs, one of these ERVs, the ERV 111, Bessie found in the HL60 neutrophil-like cell line to be expressed as mostly full length. So this is sort of the full-length ERV 111 locus. And we can see many of these reads that are actually spanning the entire locus from long read sequencing, suggesting that these ERVs are in fact being expressed as a full-length transcript in these cell types. And so Bessie, who's sitting in the back here, she's currently investigating the role of these ERVs and how they're contributing to neutrophil activation. We're going to be using some of these cells to be able to assess those going forward. So stay tuned. All right. And so, so this is sort of what we've been doing in terms of understanding how these can contribute to inflammation. In the last little bit, I thought I might share another story that we've recently published, where instead of sort of how these ERVs are important for how they contribute to inflammatory diseases, we've also shown that these ERVs are important for beneficial effects of these ERVs, beneficial effects in the context of antiviral immunity. And so, although I'm almost running out of it, is this the right time? 10 minutes. Okay, we got this. All right. So the model that we're really using is a mouse model to really investigate this. And this is a model that we're also continuing to use in the lab. So I thought I'd share this. So it had been known from others' work that ERVs can kind of reemerge in immunocompromised strains of mice. So for example, if you take some of these immunocompromised mice like RAG knockouts or MIT88 or TLR7 knockout mice, you can see these ERVs really reemerge. And in fact, what we and others have shown is that there are a number of proviral loci that are present in the mouse genome, but the two that are really important for the reemergence of these ERVs in immunodeficient mice is the XMV43 and the ecotropic ERV locus called the EMV2 locus. So these EMV2 and XMV43 locus are really highly elevated in TLR7 deficient mice, and these lead to these are the ones that are, these are proviral ERV loci are really the ones that are contributing to the reemergence of these ERVs in these mice. And so we had shown that in these immunodeficient mice, in particular the TLR7 knockout mice, we can find infectious ERV genome, and this is sort of the genome structure. In fact, what happens is that the EMV2 ERV locus combines with XMV43 locus, and this complementation can lead to generation of infectious virions. And we can measure this by taking leukocytes that express lots of these ERVs, co-culture them with a susceptible cell line, and really see amplification of these endogenous retroviruses or virions generated from sort of all over the body of these TLR7 knockout mice. And so we use sort of this model system to then ask, how does the reemergence of these ERVs actually contributing to a biological phenotype? And so we were really interested in pursuing antiviral immunity because there had been studies that had shown that ERVs can contribute to antiviral immunity in various mechanisms, whether by blocking capsid uncoating, receptor interference, or even competing with other retroviral genomes or sequences. And so we tested this in the context of HSV2, which is a sexually transmitted infection. And HSV2 is a large double-stranded DNA virus that affects over 400 million people worldwide. And really there's no vaccine for this currently. And so it's really important for us to really understand the pathogenesis of this virus in the context and novel antiviral factors against this virus. And so what we had found was that when we infect these mice, these TLR7 deficient mice that have really high levels of ERVs, and you can kind of see the ERV expression in the vaginal lumen here, stained for the ERV envelope protein here. When we infect these mice with HSV2, we found that the TLR7 knockout mice actually are more resistant to HSV2 pathogenesis compared to wild-type mice. And you can see this in disease score as well as in survival. And not only are they sort of susceptible, more resistant, you can actually take those purified endogenous retroviral virions, pre-treat wild-type mice with these virus intravaginally prior to HSV2 infection. And even sort of in that context, we saw that these infectious herbs that we pull out from the TLR7 knockout mice are sufficient to reduce disease, HSV2 disease, as you can see here, and delay lethality of these mice. And so when we further shown this in a more susceptible mouse background, like an IFNAR knockout background, interferon receptor knockout background, and even in this sort of more susceptible background, you can see that if you pre-treat these mice with these infectious virions that we isolated from the TLR7 knockout mice, they have a delay in disease compared to the non-treated counterpart and increased survival. And what we had further shown is that was sort of the sufficiency experiment. And to show that these were actually necessary, we deleted the EMV2 locus from the TLR7 knockout mice to generate a TLR7 EMV2 double knockout mouse. And you can see that these double knockout mouse no longer express ERV envelope protein on leukocytes, and there's no longer infectious ERVs that can be detected from these EMV2 TLR7 double knockout mice. And in fact, when you infect these double knockouts, they no longer have this resistance phenotype that you saw in the TLR7 knockout mice here. So if you delete this EMV2 locus, these mice are just as susceptible to HSV2 as the wild type counterparts, really showing that this EMV2, this infectious ERVs, is necessary for a sort of protection against HSV2. And we found that this was happening in an interferon independent manner. And so right now we have a number of projects in the lab that I won't get into. Many of the students that are sitting here are working on various aspects of sort of the mechanisms that lead to this novel ERV-mediated protection against HSV2. So please stay tuned. And we're also using this mouse model not only to further study HSV2, but really to understand further how ERVs contribute to immune homeostasis. And this is a project that some other students in the lab are working on, Tatiana and Phoebe, which I don't have time to get into. And finally, we're really trying to understand and use the system to be able to leverage these ERVs to modulate our immune system and to be able to treat disease. Can we genetically manipulate them? If we understand how they're tuning the immune system, can we sort of leverage that? Can we develop novel therapeutic vaccination approaches against this? And we have a new student in the lab, Erin Evans, who supervised with Mark Horowitz, and we're really also starting to get into other autoimmune diseases like MS as well. And really, this is the tip of the iceberg. There are clinical trials that are targeting human ERVs. A number of them are listed here, but I think this is a growing area in the lab, and it's quite exciting for all of us in the ERV field. And with that, I like to kind of acknowledge my lab. We started on Zoom in the middle of the pandemic, and we also had to do some even outdoor social distancing pictures back in 2021. And now we have a large lab with lots of students who are really working hard on various aspects of this research. And with that, I'd like to thank all my collaborators, all the M&I staff, the cores, and my funding agencies. And thank you for listening. Yes, Kelly.
I love listening to you talk, Maria, because it makes me think in really different ways than I'm used to. A couple of really broad spectrum kind of questions. So the ERVs, are they turned on in the thymus so you get tolerized to them? And if that does happen, does that ever make you have a hole in your repertoire so you can't respond to a virus that looks like it when you get infected? So that's on the one side.
Okay. Yep.
On the flip side, viral infections frequently come along with baggage like autoimmune disease. So if you get a viral infection and it has a close enough mimic to an endogenous retrovirus, is that what kicks off autoimmune disease like arthritis and MS? And, is that?
Yeah. So really amazing questions, like something that we do think a lot about. And so I guess there are a couple of components there. Are they expressed in the thymus? This is actually something that people are working on now. We just came back from a keystone for transposable elements, and there are a handful of groups kind of really trying to dissect that out. And there is some elements that are expressed in the thymus, but sort of the comprehensive nature of like which herbs are expressed in the thymus and which herbs are not. Are they all expressed? Or are they not? Sort of that level of resolution we don't have yet, right? But that being said, we know that you do have, you can get in context of disease, there are T cells and B cells against these herb antigens. And so, some of this, there is some escape of, even if they are expressed in the thymus, there is some escape of that tolerance, right, on the periphery that we can see in disease context. And sort of, does that, if there are some eliminated, cells that are eliminated in the thymus, can that actually cause a problem later on to fight against other retroviral infections? I mean, that's really interesting. I don't think anybody has looked at that, and whether there's cross-reactivity against sort of herb immune surveillance of herbs versus things like HIV, for example, that hasn't really been shown that there is cross-reactivity. So I don't know if the sort of lack of recognition of herbs or can contribute, leave a hole, right? And on the other hand, viral mimicry of these herbs and some of the other viral pathogens, that's also a whole new area that really is understudied. And really there hasn't been distinct viral mimicry that has been established within the herb repertoire that we're studying. And so all sort of open, sort of ended questions that are really important and interesting to study, right? Yeah, sorry, I don't have many answers, but.
I just have a question from someone through Zoom. And they say some herb research looks for relations among variants within herb sequences and health and disease outcomes. Have you done any of this work or have any interest in pursuing it?
Yeah, so in terms of differences in actual sort of SNPs or variants of herbs that are expressed between healthy and disease, we haven't touched that. There is interest in sort of potentially herbs that are insertionally polymorphic between individuals, or sort of these really recent herbs that are still insertionally polymorphic between individuals. Those could be different between disease and healthy, and that's an interesting area that some groups are pursuing. We haven't kind of explored that area, but if there's anybody who's interested in looking more into it, I'm happy to sort of collaborate. Yeah.
Very interesting talks. I was curious to know whether the expression of herbs can be driven or by line one elements, like carried along.
Yeah. So what we think, how these herbs how are they regulated, right? How do they get expressed? So what we know is that they are epigenetically silenced. And so what actually ends up leading to expression of these herbs is release of epigenetic silencing, in particular by, for example, things like crab zinc finger proteins, which are DNA specific, or LTR specific crab zinc or finger proteins that can really recruit chromatin silencing marks to these sites and lead to sort of establishment and maintenance of epigenetic silencing at these herbs. And when those are sort of relieved, then we see induction of herbs and transcription of herbs. And so downregulation of things like TRIM21 or SETDB1, all of these sort of silencing machineries that are sort of important for establishing a maintenance of silencing of herbs. When they're released, then you get herb expression. Whether lines contribute to that directly, I don't think that's been shown. We think that sort of herbs and lines can be dysregulated when there's a lack of epigenetic silencing, right? But they seem to be working in a slightly different mechanism. So yeah. Hey, Selena.
Around in the first part of your talk, you had looked at the envelope gene specifically, and you said it was common to a lot of those elements. Was GAG also common to those elements? I think we've talked about this briefly before.
So GAG is also common to many of these proviral sequences. At the time, we didn't specifically look at GAG. We were looking at anti-envelope antibodies, but certainly GAG is expressed and present in a lot of these herbs, and they could potentially be serving as additional sources of antigens, intracellular antigens that we would like to pursue later. So I mean, I think the library or repertoire of herb antigens that we could potentially explore as targets for in the context of autoimmunity is large.
Yeah.
We haven't gone there yet, but that's something to think about in the future.
And just quickly, do you think that there's some kind of like threshold effect happening here where like chronic herb expression is leading to like higher thresholds for antiviral responses and things like that? And then when you have another insult from another viral infection or something, then you've sort of moved things off. And is there any studies that sort of compare like different levels of herbs, chronic stimulation of herbs?
I mean, I think that's what we would love to be able to show. No study has done that, but that's sort of the reason why we're interested in this, right? Can the virome tune, where our immune system stand, and can that dictate where you end up in terms of how you respond to viruses or how you develop disease? No one's really kind of done that direct relationship, but we would love to get there. I think other members of the virome, it's been shown to modulate response against bacterial infection, things like that. But for herbs specifically, we haven't gotten there yet, but very interesting.
Yeah, very interesting talk. So most of these herbs, do they express functional proteins or dysfunctional ones, which are probably right away degraded and end up in the lysosomes?
A lot of them have mutations, right? So they're not going to be able to generate full functional proteins, some of which can. So I think those are the ones that we're really focusing on, these proviral herbs that have still maintained intact open reading frames that have the coding potential. And for example, the envelope proteins, they don't have any mutations. They are full open reading frames that can be that have the potential to be translated. One of the projects in our lab is really trying to do a comprehensive analysis of sort of the translatome of these herbs that are actually transcribed, because I think I don't think we have a good sense for how many of these herbs are actually producing translated products. That's something that that is important in the field to understand. But we can see, at least from in silico, which are intact open reading frames and which are not, right?
Do you know if some of the herbs are transcribed in the antigen presenting cells, APCs?
Yeah, so even in endogenous cells, there are different cell type specific expression of these herbs that we and others have seen in different immune cells. So and certainly there are T cells that are, for example, in cancer patients, there are T cells that are specific for herb antigens, and that can also tell you that these herbs are actually being presented by antigen presenting cells and leading to T cell responses, right? And so certainly I think the distinction is sort of, are there herbs that are important in the physiological function of these immune cells in and of themselves, and are they actually being used as antigens to prime the immune system? And I think they're both really interesting questions that we're interested in pursuing.
One more online.
Okay. Sorry, we're running.
Do you feel that individuals with HERV-associated diseases such as MS can be blood donors? Different approaches to donor eligibility with MS are taken by different blood operators. For example, American Red Cross accepts, but Health Canada does not accept on the basis of risk of an inferred transmissible agent.
So the question is,
do you feel that individuals with HERV-associated diseases such as MS can be blood donors?
Blood donors. Is that related to sort of the research? I think this is kind of a bit off topic. Maybe we can discuss more offline.
Yeah.
But yeah, thank you.
Yeah, so interesting talk. For COVID-19, neutrophils have been shown to be activated and produce nets. So I'm wondering if you're thinking that these herbs that you found that are associated with COVID-19, if they might induce nets through similar or different mechanisms.
Yeah, so this is a sort of different paths that we're really interested in taking. Are the neutrophil activation, sort of the correlation that we see between the herbs and neutrophil activation that we for COVID-19, similar to what we see sort of in the lupus case, where there are some autoantibodies against these herb proteins that can then contribute to sort of this net formation through immune complexes via immune complex generation. That's sort of a separate project. We do have COVID-19 serum that we're that in our freezer, and we're just kind of figuring out, a good time to be able to assess herb reactivity against or reactivity in those samples to look at sort of antibody dependent net formation. Aside from that, though, what Betsy is working on is really trying to understand, do these herbs that are potentially expressed in these cell types, do they have physiological functions within these cells that might contribute to differential activation status, right? And so we're going to do some loss of function and gain of function experiments to be able to get at that.
Thank you.
Yeah. Yeah, thank you.